Hello, welcome to my YouTube channel and please subscribe. Um, Today we are going to be talking about the classical pathway. So, the classical pathway is a chain of events in which complement components react in specific sequences as a cascade resulting in cell lysis. It is activated by antibody bound to antigen, forming an antibody antigen complex, but never by native or free antibody. These complexes may be soluble or they may be formed when an antibody binds to antigenic determinants or epitopes situated on viral, fungal, parasitic, or bacterial cell membranes. Soluble antibody antigen complexes are often referred to as immune complexes and only complexes formed by IgM or certain subclasses of IgG antibodies are capable of activating the classical complement pathway. Activators of classical pathway include we have the immunoglobulin IgM, and IgG. Subclasses may vary with regard to their efficiency in activating the complement. So we have IgG3 are the most efficient, followed by IgG1 and IgG2. So you have to note that IgG4 does not activate the classical pathway. So we have the staphylococcal protein A, it's also an activator of the classical pathway, C reactive protein, and DNA. Steps in activation of classical pathway. The classical pathway of complement activation usually begins with the formation of soluble antigen antibody complexes or with the binding of antibody to antigen on a suitable target, such as a bacterial cell. So the initial stage of activation involves the complement components C1, C2, C3, and C4, which are present in plasma as zymogens. So the formation of an antigen antibody complex induces conformational changes in the non-antigen binding portion of the antibody molecule. This conformational change exposes a binding site for the C1 component of complement. This, as a result, results in sequential activation of C4, C2, and C3. In serum, C1 exists as a molecular complex consisting of one molecule of C1q and two molecules each of the serine proteases C1r and C1s, which are held together by calcium stabilized complex C1qr 2s2. The C1q molecule itself is composed of 18 polypeptide chains that associate to form six collagen-like triple helical arms, the tips of which bind the CH2 domain of the antigen-bound antibody molecule. So each C1 macromolecular complex must bind its C1q globular ends to at least two Fc sites for a stable C1 antibody interaction to occur. C1q binding in the presence of calcium ions leads to activation of C1r and C1cs binding of C1q to the CH2 domains of the Fc regions of the antigen complex antibody molecule, which induces a conformational change in one of the C1r molecules that converts it to an active serine protease enzyme. So the two C1r proteases then cleave and activate the two C1s molecules. So activated C1s is an esterase which is going to split C4 into two fragments, a small soluble fragment C4a and a larger fragment C4b. C4A has an aflatoxin activity, and C4B binds to cell membrane along with C1. C4B, in the presence of magnesium, splits C2 into C2A and C2B. So the smaller fragment C2B diffuses away while the larger fragment C2A remains attached to C4B. The resulting C4B2A complex possesses enzymatic activity and is called a C3 convertase, which converts C3 into an active form. So the C3 convertase activates thousands of C3 molecules and splits these molecules into C3A and C3B. A single C3 convertase molecule can generate over 200 molecules of C3B, resulting in amplification at this step of the sequence. So the biological importance of activated C3b as well as C4b is that they are able to bind C3b stroke C4b receptors present on almost all host cells, most notably phagocytes. The increased affinity of phagocytic cells for C3b, that is C3b, C4b coated particles, is known as immune adherence. So, immune adherence is responsible for a significant enhancement of phagocytosis, which is one of the main defense mechanisms of the body. Some of the C3b binds to C4b to A to form a trimolecular complex C4b to A 3b called C5 convertase. The C5 convertase splits C5 into C5a and C5b. So we are going to have C5a diffusing away while C5b attaches to C6 and initiates formation of C5b9 complex, which is known as the membrane attack complex. And with this, we come to an end of the classical pathway review. And I want to thank you for listening.